So the first chapter in bio for SPM would be chapter 8, classification system. Wait, no. Then the chapter's name is called biodiversity. So 8.1, classification system and naming of organisms. So this naming system is called taxonomy, this naming thing. So it involves the classification, identification, and naming of organisms. So yeah, it involves three separate parts: classification, identification, and naming. So for classification, organisms are categorized based on physical features in the taxonomic hierarchy system. For identification, they are identified using the dichotomous keys, and they are named using binomial nomenclature. So we will go to them one by one. Wait, I'm not so sure about classification. Taxonomic hierarchy. Oh yeah, that's the kingdom stuff. Okay, so first of all, we want to classify the organisms based on their characteristics. So there are 6 kingdoms: Archaebacteria, Eubacteria, Protista, Fungi, Plantae, and Animalia. So Animalia, sorry. So I'm going to skip this part because they mentioned the whole part in here. The first thing is Archaebacteria. So Archaebacteria is a prokaryote and it is a unicellular organism. So what prokaryote means is it has no real nucleus. What no real nucleus means is they don't really have a nucleus with a membrane. They don't have a nuclear membrane. They have like DNA. Some of them do have DNA. Some of them do have like other stuff that you normally see in the nucleus, but they don't actually have an actual nucleus with a membrane. It's like embedded. So we call that prokaryotic. They have no real nucleus, and they are primitive bacteria. That means they haven't really evolved much. They have cell walls with no peptidoglycan. So peptidoglycan is the reason why they are archaebacteria instead of eubacteria. So we'll get more to that later. And then the main thing about archaebacteria is that they are very good at adapting to their surroundings. They are only found in these very extreme conditions. So for example, methanogen, halophile, and thermophile. So methanogens come from the word methane, and it is for anaerobic bacteria that secrete methane as a metabolic product. So the reason why they secrete methane, I'm actually not so sure, but they are able to survive in very hard to breathe locations. They don't really breathe. They don't really need to depend on breathing that much, I guess. But yeah, that's what it means by anaerobic bacteria. That means they can breathe by themselves. You don't really need to be exposed to oxygen that much. So they can survive in places where it's very hard to breathe, like swamps, because swamps are very muddy and it's full of a lot of impurities. And the digestive tract, that thing is disgusting as hell. So next one is halophile. So halophile is halo means salt. So phile means like, so like salt. So archaebacteria that are halophiles like salt, and they live in very high salt condition areas. So that is including like mangrove, of course called wetlands, like mangrove areas, and the Dead Sea. And then there are also thermophiles. So thermophiles are people that like high temperature, not people, bacteria that like high temperature. So like how most bacteria need to survive in like a more room temperature level, but these thermophiles, they can survive up to double that or more than that. So for example, the Yellowstone National Park. Next is Eubacteria. So Eubacteria is also a prokaryote. Yes, no real nucleus, but it is a true bacteria, which means that it has cell walls made out of peptidoglycan, or in this case murine. So murine is a polymer made out of sugars and amino acids. So you don't really have to know what peptidoglycan is made of. You just have to know that eubacteria have peptidoglycan. They are obviously unicellular, and eubacteria have some of these organelles like ribosome and plasmids, but as they do not really have a real membrane, like they don't have nucleus, for example, they also don't have any other membrane enclosed organelles, which means that they only have one membrane. So yeah, that's the main thing. That's the main reason why they don't have a nucleus, because they don't have a membrane inside of the membrane. They only have that outside membrane that they have, the cell membrane, but they don't really have these plasma membranes, these membranes of organelles. So that means they also don't have those organelles such as mitochondria, endoplasmic reticulum, and others. I think that includes like the Golgi body. I'm just spitting names that make sense. So yeah. They have ribosome and they have plasmids. Remember this. They have ribosome and plasmids, but they don't have anything else. And then bacteria are classified according to the shape. So for example, they have like spiral, they have cylindrical. I don't know any other stuff, to be honest. I haven't read about microorganisms in a while. Next is protista or protists. So they are eukaryotic. That means they have an actual nucleus. And they can either be unicellular or multicellular. So unicellular might include Chlamydomonas. Chlamydomonas is a protist. And for multicellular, what are? I'm not so sure on that one. I think Spirogyra is. Wait, let me see. But they're both algae lah. Algae are a type of protist. And other types include protozoa and slime mold. So slime mold, they don't really mention much. Might be worth just knowing one of the examples. But for protozoa, the main ones that you have to know is Paramecium and the one that causes the one that causes malaria. But which I'll get to later. But yeah, the eukaryote thing relates to this. They have an actual nucleus bound by a nuclear membrane, and they also have other nuclear membrane bound organelles like mitochondria. And these algae, they have chloroplasts. Not all of them have chloroplasts, but yeah, they'll mention more about this later. And then the next one is fungi. Fungi is probably the most important one in this chapter. So it is eukaryotic. It can be unicellular or multicellular. For example, unicellular would be yeast. It says here yeast. And then mushroom would be the multicellular type. And they are heterotrophs. Heterotrophs means that they cannot produce food by themselves. They have to obtain food from somewhere else. And the main difference about them and other types is that they have a cell wall made out of chitin. So some like plants have cell walls made out of cellulose, and others have cell walls made out of peptidoglycan. But fungi have cell walls made out of chitin. So yeah. And another thing is that they have hyphae, right? We know about the hyphae, like Hydra stuff. We've learned about hyphae before. Hyphae. And this network of hyphae is actually called the mycelium. You know the Minecraft mycelium? Yeah, it's basically a network of if you go to those large-scale fungi areas, they're all connected down there, and they are connected underground through hyphae, and the whole thing is called mycelium. That's why in Minecraft you have those like mushroom islands, and then they're called mycelium. That's basically the difference is that most of these are underground. Next is Plantae and Animalia. So Plantae, I'm sure these are all quite obvious lah. These are all something that you don't really need to, you don't really need me to explain. We've learned about them a lot. Okay, next is taxonomy hierarchy. So how do we classify them? Like how do we classify them more specifically? So yeah, we're still in classification. So how do you do that? You use something called the lineal, I think it's called lineal hierarchy system. I don't know if it's called linears. I just call it the lineal hierarchy system. And they classify the things according to their hierarchy, like what rank they are. So here's the hierarchy system. It starts with the domain. So domain is either prokarya or eukarya. So either prokarya or eukaryote lah. And then next is kingdom. So either in the animal kingdom or in the plant kingdom. And then after that, they go to the phylum, class, order, family, genus, and species. So yeah. So the highest rank is domain, and then it gets more specific as you go down the rank. So domain is the least specific, it's the most general, and the most specific is the species, which actually determines what it actually is. So for example, Panthera leo and Panthera tigris. So I'm sure you guys know what those are: lion and tiger. They come from the same genus. They come from a similar family, the panthers or Panthera. But then they are different types. So they are different species. So yeah, we'll talk about more about species later. But just saying. You can identify when two organisms are of the same species when they can crossbreed, when they can reproduce. Next is, how do you name this? you now know how to classify them according to the hierarchy system. But how do you give them a name? You can't just give them a whole name called Eukarya, Animalia, Chordata, Mammalia, Carnivora, Euclidae, Ursus, Arctos. So instead, we use the Linnaeus binomial system, which is to name them based on their genus and species together, and these names are based on Latin, and they must be italicized or underlined if you're writing them down. So yeah, that's why when you see these these Latino names, these scientific names, they're always italicized. That's just how it is. But yeah, it's the genus and then the species. For example, Panthera leo. Panthera is the genus and Leo is the species. So yeah, there's not really much that I can say here. I hope none of these come out because this is very hard. This is not worth memorizing. So yeah, the reason why sorry. The reason why we need to name this is because we need to name this according to this binomial nomenclature. Is because different countries or different regions name things differently. For example, some call it corn, others call it maize. So instead of calling it corn or maize, which will cause confusion, they give it a scientific name, and that's the same for everything else. Next is dichotomous key. So if trying to name a species is using binomial nomenclature to classify multiple species into their own specific types, is called dichotomous key. And they use something called couplets, where you try and ask yourself, what's it called again, an inquiring question. Where the answer is either yes or no. So, or trying to divide that into two types. That's called coupling. So dichotomous key, di means two. That's why you want to use two types. Is it an animal or is it a plant? Does it have legs? Yes or no? Does it have more than one pair of legs? Yes or no? And so on. And it goes down. So it's basically one or two. If it's one, is there only one animal left? Then if it is, then you choose that one. If it's two, and then like, is there any other animals that are there multiple animals that are like two? Yes, then go and separate them into. So is it 3 or 4? If it's 3, then blah 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 blah. You just continue and until you get all of them into their own types. So yeah, this is the more common one. This is the more common dichotomous key. I don't know what's actual name. This is the other one called the spider key, but it's the one that we normally use. Next is biodiversity. So there are 3 types of biodiversity: genetic, species, and ecosystem diversity. So genetic is like the gene variation of an individual within the population and between populations. Like for example, humans. For example, humans in Malaysia, I won't talk about skin. I'll talk about height. So I am considerably tall in Malaysia. Not considerably tall. I'm averagely tall in Malaysia. But if and not everyone is as tall as me, and some people want to be taller than me. So that means that there is a genetic diversity because some people are tall, some people are short, and other people are average. And then if I compare myself, a Malaysian, to maybe someone from America, Americans are quite tall. They are like the average height there is like 10 or 20 cm more. So that shows that there's a difference between the genes of Malaysia and the genes of the people from the US, for example. So yeah, so to make sure that you get genetic diversity, you want to make sure that no two individuals are identical. Not even twins. Like maybe twins can even have some different face shapes even though they are identical. So yeah, that's genetic diversity. It says here paddy. There's like a lot of types of rice in the world basically. Okay, next is species diversity. So species diversity is how many types of species there are in the community or in the habitat. So it includes the total number of species in the community and the species distribution. So how many species are there, and are they evenly spread or are they more clumped up together? So yeah, the more species you have, like for example, maybe your ecosystem has like grass, maybe it has spider, maybe it has mosquito, maybe it has bird, and so on. The more you have, the more the more it has species diversity. And then lastly, it's ecosystem diversity. So it refers to the biotic community and ecological process in ecosystems on the land, sea, and other aquatic environments. So that includes what ah? It's similar to species diversity where where it involves the biotic community, because species are part of the biotic community, but there are like other factors, I guess you can say. It involves if species diversity refers to like the I won't say animals lah. Yeah, the species themselves. Ecosystem diversity refers to like the whole habitat or the whole ecosystem. So yeah, it combines all of those factors together. Next is phylogenetic tree. So phylogeny is used to show the evolutionary history of a species or a group of organisms that are genetically linked. What that means is a phylogenetic tree is used to show how much has a certain characteristic evolved over time. Like maybe back then we were related to fish, but now we're not. And maybe fishes are, and then fishes like and maybe some fishes are related to each other, but now they're not. And so on. So you want to, so this phylogenetic tree is to show how have they changed over time. How have these these species evolved over time? How did they differentiate from each other? So you use something called a homologous structure or same structure, and it refers to like one certain characteristic that they have uncommon and try to separate them based on that. Like for example, normally in the exam you'll see a phylogenetic tree like this. So think of it as like this. So yeah. So this is what you will normally see in exam, where there is like what's it called again? I forgot what it's called already. It's like it's a terminal and a node. If I'm not wrong, this is your terminal, and these are your nodes. If I'm not wrong, go read the workbook more, and do it yourself. Do the workbook. The workbook is really good. But yeah, if you don't want UEC, okay, I'll say if you don't want UEC, if you don't want to read UEC, just read the workbook. The workbook has a lot of UEC related questions, so that's why I kind of like it. But back to this. This is what you will normally see in exam. Instead of this like square divide thing, it will look more like this, like an F. And what it shows is it starts from here, and this shows like the beginning, like the oldest ancestor. And then normally here you will write like the characteristics of a species. Like for example here, they have like here, they have vascular bundle. So before that, the ancestor, or which is algae, does not have any vascular bundle. And then suddenly some of them evolve the other way. Some of them evolve in one way, which is they remain not having vascular, but others continue the path of having vascular. But others evolve and have vascular bundles. So now they separate. And then from here, after the vascular bundles either have seeds or no seeds. So if they don't have seeds, they go here. If they do have seeds, they continue the path. And then if they have seeds, do they flower or do they not flower? So if they flower, they go here, and if they do not flower, they continue down, and so on. Okay, for this one, it mentions again like in another topic, so I won't talk about in situ and ex situ yet. But I guess I can mention a bit lah. Long story short, in situ conservation is to maintain species in their natural habitat, while ex situ is more towards zoos, like taking them out of the natural habitat to keep them safe. So they have their respective advantages. For in situ, the animals get to live in their own habitat, but for ex situ, the animals are able to be more observed instead of like getting hunted down. Next is microorganisms and viruses. So yeah, what are the main characteristics? Normal bacteria, as you mentioned earlier, eubacteria and archaebacteria both do not have nucleus. So they are prokaryotes. And they do have genetic materials. They have something called a nucleoid, but they don't have a real nucleus. So they have plasmid, they have ribosomes, but they do not have mitochondria, they do not have endoplasmic reticulum. The size, I think that's normal. And then they can exist in a single cell, diploid, filaments, chains or clusters. And then they can exist in a few shapes, like the spear, coccus. So coccus is like balls. Vibrio, so it has a comma shape. Bacillus, which is cylindrical or rods, and spirillum, which is just spiral. Next would be protozoa. So protozoa, they are unicellular, but they kind of act like animals where they can move. They can move, but how do they move? They don't have feet. They use something called pseudopodia. They either use pseudopodia, cilia, or flagellum. Flagellum is like a tail that they have here. This is the bacteria's tail, but protozoa have it more often. So this tail is to help them move around. So like how like the fish flaps its gills. It's not gills. It's what's it called again? I forgot. Oh my god, I'm so bad. Its tail. So that when the fish flaps its tail gun, it will like cause movement. That's what the flagellum is for. And if not, they can use cilia. So cilia is like very sticky legs. And then like the legs continue sticking and sticking and sticking. So yeah, it slowly moves. And then it has pseudopodia, which is false feet. So like I think what it does is it accumulates in one region, and then it slowly like what happens is they accumulate in like one region, and then it suddenly combines like this. Like so it will move towards here and combine, and then in the end the protozoa will move in front because it has to pull. It's pulling the protozoa. So yeah, some of them can undergo photosynthesis. So an example would be Eugulina, while others are heterotrophic, which means that they need to find food. Next is algae. So algae, again, they're either unicellular or multicellular. And some algae also have flagellum, the tail. And the thing is that algae have chloroplasts, so they can actually undergo photosynthesis for themselves. Next is fungi. So fungi is like the most interesting one because it's the most different, the most unique. It is heterotroph. It is either parasitic or saprophytic. I'll talk more about those later. And they have chitin walls, and then they have like inside here are like hyphae. And then they connect together, and that's called mycelium. And yeah, most of these I already mentioned. Next is viruses. So viruses aren't actually living things. That's why they're not included in any of the kingdoms. And that is because they cannot undergo cellular processes by themselves. They need to connect to a host first, and then they will do its stuff. Like for example, COVID can't survive on its own. If you leave COVID alone for a few days, it will just die. It won't do anything. But once COVID finds a host body, it will start living. It will start doing its processes. So basically viruses cannot reproduce. They cannot excrete waste. I think they cannot breathe. But once they actually find a host, they can duplicate through that host. That's really scary. So because of that characteristic, they are not living things. But they do have nucleic acid, DNA or RNA, and they have capsids. So the capsids are protein. The whole thing is called vitriola. I forgot really, but it's not really worth knowing. These are quite obvious. They are very small, and examples include this. Okay, next it is I would say a fan favorite in exam. So this is like a favorite question: nitrogen cycle. So I'm gonna try and explain this in the best way possible. First of all, our atmosphere contains more than 70% nitrogen gas. So how do we But then these plants, they cannot directly absorb the nitrogen for the air. So how do we turn this nitrogen into something that can be absorbed by the plants for their growth? First of all, they need to be delivered back into the soil. So there are two types of ways where they can be delivered back to the soil: natural method or unnatural method. But both of these are called nitrogen fixation. So that all three of these are nitrogen fixation. Nitrogen fixation. So what is nitrogen fixation? It is the process of converting the nitrogen in the air to become nitrogen in the soil. So one example would be atmospheric fixation, where the nitrogen in the air will react to the oxygen during these like storms to become nitrites, and then when it rains, the nitrites will go into the soil, and then they'll end up going here lah. Next is legume root nodules. So what do the legume root nodules do? Well, they don't do anything, but these legume root nodules actually contain something called rhizobium. So rhizobium here, they basically they absorb any nitrogen gas that they can find. They absorb it, and then they convert it into ammonia, and then the azobacter converts it into ammonium. So yeah, that's kind of specific lah. But you can just say that rhizobium and azobacter convert the bacteria into ammonium. Just say that. But if you want to be more specific, rhizobium converts it into ammonia, and azotobacter converts it into ammonium. But all you can just say is nitrogen fixation. And the last one is the unnatural method, or the what is it called again? Man-made synthetic method, which is through fertilizer, where they just use nitrogen in the air and then they process it to make either nitrate ions or ammonia ions. So if they're ammonia ions, they will actually go here. But if they're nitrate ions, they can be immediately used by the soil. But yeah, that's the thing. All three of them are called nitrogen fixation. And then when you die, you contain some nitrogen. So the bacteria or fungi will decompose them into ammonia or ammonium. And then after they decompose it into ammonia or ammonium, then the nitrosomonas will further decompose them into nitrite ions, and then the nitrobacter will convert them into nitrate ions. Remember to put the SP. The SP means species. You have to put it down. I'm just not saying it because it's easier. So yeah. Rhizobium and azotobacter are for ammonium, but then to turn the ammonium into nitrite, you need nitrosomonas, and to turn the nitrite into nitrate, you need nitrobacter. So this process is called nitrification. And then after that, when you get the nitrate, the only thing that plants can absorb is nitrate ions. And then after that, if there's any leftover nitrate ions, some bacteria will denitrify it, so it's called denitrification here, and then they will send it back to the air, and then the process repeats itself. Trust me, this thing should come out. It's something that people really love. So I hope if you want, just repeat this part. Next is the role of microorganisms. So what can microorganisms do? First of all, they can be producers. So these ones are usually the ones that are autotrophic, which means they can make their own food. So they will start the food chain. Next is decomposers. So some microorganisms like fungi and bacteria, they can actually decompose. They can decompose these materials into simpler compounds. Like for example, in here, they decompose these complex compounds into some things like ammonium or ammonia. So what happens is, these decomposers actually secrete enzymes, and then the enzymes break down the organic materials, and then after that they break it down into simpler materials, and then they will absorb it. Next would be yeah. So yeah, they don't just make ammonium, by the way. They can also make carbon and sulfur. Next is parasites. So parasite in general means one organism takes over a host, but one person takes over a host, but it will harm the host instead. So instead of like it helping the host, it will harm the host while itself gives benefits. So the most popular example would be Plasmodium species. So Plasmodium species is actually a protozoa. This is the example that I mentioned earlier. And the Plasmodium species lives inside mosquitoes, called specifically the Anopheles mosquito. And this mosquito, when you get bit by it, may cause malaria. And lastly, which is I think the most important one, will be the symbionts. So symbionts are organisms that have close relationships with other organisms, and when these two have close relations, it is called symbiosis. So there are two types of symbionts. One is ectosymbiont, which is like outside the host cell, and endosymbiont, which is inside the host cell. I think you can this yourself. Ectosymbiont fungi outside around plant roots, and endosymbiont protozoa in termites, like inside termites. Next is pathogens and vectors. So pathogens are organisms that can cause diseases, like for example viruses, bacteria, protozoa, and fungi. So bad stuff. And I don't have to elaborate this further. Next is vectors. So vectors are the types or are the organisms that transmit pathogens. For example, Vibrio cholerae will spread cholera. And then next is Aedes aegypti will spread dengue, and Salmonella typhi will spread ringworm, I think. So I think it's called ringworm. I forgot already. You know that's tinea. So that's fungi. So I'm guessing that this will spread Salmonella. So, but yeah, the most popular one will be Vibrio cholerae and ringworm. So there are a few types here. For virus, hepatitis B, bacteria, TB, protozoa, dysentery, and fungi, tinea versicolor. Yeah, there's a type of tinea called ringworm. That one is more common, ringworm. So